Hi everybody, my name is Energetic Care and today I would like to give you guys a Cirm update from my host lab at UCR from Dr. Prue Talbot's lab. So before we dive into what I do for my project, what is my project? So I study the toxicity of e-cigarette constituents in human prenatal development. So before I start talking further more about the project, I want to talk about some background information on to why we wanted to do this project in the first place. And it's because there are pregnant women that are getting exposed to vape aerosol products and that are vaping or not vaping that can affect the development of the embryo, as well as study the metals zinc and chemicals like methylglyoxal that is detected in the aerosol and the fluids in the electronic cigarette. In other words, what are the chemicals doing at the human prenatal stages? Not only is this important to look at how these processes are affected in development, but stem cell population that exists in adults can be affected similarly. And this is because stem cells are important for our organs. This bridges the developing human and the adult human together. And as for our overall goal, we want to ultimately see the effects of certain key chemicals and metals that are in electronic cigarettes on embryonic stem cells and see how treatments can affect them in the long run. And this, we will first be using the trilineage differentiation kit to get the primitive three germ layers known as the endoderm, mesoderm, and ectoderm. And we also want to see what effects they have on rate of differentiation. Meanwhile, in the long run, we want to see what the H9 female cells can get to the neuro-ES cells and to differentiating neurons. As for our hypothesis, we hypothesized that upon testing the toxicity, you might get more astrocyte differentiation when you treat the embryonic stem cells with the chemical methylglyoxal, and even get maybe fewer neurons and more cognitive problems. Astrocytes are the most abundant cell type in the central nervous system, which are rivaling the diversity of neurons in cellular morphology, gene expression profiles, and developmental origins. And furthermore, we also hypothesized that upon testing the metal zinc, it would slow down the differentiation process. And this is because zinc ion acts as a time dependent slow inhibitor. So why are we interested in studying methylglyoxal? And this is because methylglyoxal is an aldehyde which is highly cytotoxic and reactive, and it is a cell permeable and a byproduct of many metabolic pathways. And most of the methylglyoxal is actually generated in our cells, which form as a byproduct of glycolysis. And because methylglyoxal is so toxic, the levels need to be tightly regulated in the cell. And this accumulation of methylglyoxal can actually result in apoptosis. And along the lines, when further research conducted by such researchers, it has shown that methylglyoxal has many implications in degenerative diseases like diabetes, and that actually has the formation of advanced glycation end products. And for those reasons, we wanted to see how this chemical is actually affecting at prenatal stages. So, how exactly is methylglyoxal formed in our bodies? Methylglyoxal is mainly formed via spontaneous degradation of DHAP and G3P. It is also formed as a byproduct of formaldehyde metabolism and protein metabolism. Our cells eliminate methylglyoxal via the glycolysis system. Less than 1% of the total methylglyoxal exposure comes from food and drinks. Before committing to a differentiating experiment, it is wise to get some preliminary data as proof of concept and to determine what concentrations to use for treatments, which is known as our null. So we can do that by treating with metals like zinc and reaction products like methylglyoxal in different assays. So possible assays that we could look into carrying out through our project is doing an MTT for our H9 cells, possibly doing a seahorse on the H9 cells. And for the seahorse, we would be measuring the oxygen consumption rate and the extracellular acidification rate of live cells in multi-well plate, which is typically a 96-well plate. And we would see key cellular functions such as mitochondria, respiration, and glycolysis. And furthermore, along the lines, we will be doing a trilineage kit that I will be talking about as our first preliminary experiment we did. And further along the lines, once we obtain good data, we will be using treatments for our cells using the trilineage kit. And possible other preliminary experiment, we could try a neutral and a pulmonary differentiation without treatments. As for the preliminary experiment, I mentioned before that we did the trilineage kit, and with this, you can see that here is a design we use for our preliminary data for the trilineage, showing two different plates on a 96-well plate, and one plate that contains the ectoderm and control, and the other plate that contains the endoderm, mesoderm, and control. And the reason why we had two different plates was because the ectoderm, mesoderm typically takes 5 days to expand in the respective medias, and the ectoderm takes 7 days in the respective media. So to avoid any kind of problems with expansion, we wanted to plate them on two different plates. Before the terminal differentiating project, it's better to get preliminary data, like I've mentioned before. Here we are going to use the trilineage kit that is meant to look at whether the pluripotent embryonic stem cells can even make the different germ layers. We want to use this to see if the chemical methylglyoxal affects differentiation of the 3 germ layers and how the metal zinc affects the 3 germ layers specification, because it's good preliminary information to have to carry out further analysis. So as for our experimental approach, we had maintained 2 passages of H9 cell line, and we used them for the trilineage kit, and we labeled the 3 germ layers after the respective days of completion for the expansion with 2 antibodies markers for each line to look at the rate of differentiation and to see what markers work for them. So the markers to validate germ layer identification, we use ectoderm for PAX6 nestin, and for mesoderm we used BRAG-T and CXCR4, and for endoderm we use SOX17 and FOX82. And then we also used a biostation, which is basically a microscope that takes time-lapse images on its own, and you set the time, date, and what day you want your experiment to be completed and how many pictures you want it to take throughout those period of time. And from that evidence, we can see how the morphological changes are occurring and to see what other further analysis we want to do along the line. Here's a video that I would like to share with you guys from the biostation for the mesoderm that I accumulated pictures over the 5 days and later put in a software to image them and put them into a video so I can see how there's morphological changes happening amongst the cells and they come together in a monolayer form and make that diamond shape with the cells, and that definitely verifies that we are getting the respective germ layer. And further along the lines, by using the amino stain, we can say that we are definitely getting the respective germ layer and to further validate that we can use this tri-lineage kit. Thank you so much for hearing what I've been doing for my Cirm internship furthermore, and I'll continue to update you guys. Thank you and take care.